Hey, good afternoon. My name is Joseph Spears. It is the 8th of April, 2022. Just got back in, and I'm going to go ahead and jump right into our case connections for Unit 4 here. So our first one, a newborn girl appeared normal at birth, but within 24 hours she developed lethargy, hypothermia, and apnea. The attending physician suspects a metabolic disorder and orders a panel of goods. A full blood work was ordered, and this female's data is presented below. The initial blood ammonia level was elevated at 314 units deciliter and rapidly rose to 600. Discuss the normal mechanisms of ammonia transfer in the body that should be occurring, including glutamate dehydrogenase, glutamine synthase, and discuss role transamination. So ammonia is a byproduct of metabolism of things like amino acids, and other substances, and it is toxic. It is toxic to both neural cells as well as musculoskeletal cells, and different organisms have different ways of getting rid of it. Fish just secrete it directly. Us mammals, being a little bit more complex, we have to convert it into something else and then get rid of it. So that process is basically we take ammonia, producing the cells and everything else like that, gets converted into urea, goes through the bloodstream, goes through the liver, and then is excreted by the kidneys along with urine. So ammonia is produced through the metabolism of amino acids, other compounds, and again, the body has to rid itself of it. So this process for this is transamination followed by deamination. So transamination is where the amino group is removed from the amino acid, and transaminase is then transferred via glutamate dehydrogenase to alpha ketoregulate, and that makes glutamate. And then from there, the ammonia is transferred via glutamine synthase, and this produces glutamine with a reaction of basically ammonia plus glutamate. Glutamine is then transferred to the liver, and then glutaminase in the liver catalyzes the reaction of glutamine and H2O, which is glutamate and NH3. Or urea. And then that, along with carbon dioxide, that produces urea along with free ATP and water, and then that is excreted by the kidneys. So our next question is, how is nitrogen transport connected to gluconeogenesis? So in the fasted state, you may not have carbohydrates available, so like a normal source for glucose. And may need to use amino acids. These are certain types of amino acids called glucogenic amino acids. And through the process of gluconeogenesis occurring in the liver, we can turn those amino acids into glucose. So they're converted from glucogenic amino acids to alpha keto acid and then glutamate, and ultimately pyruvate and then to glucose. So our next question, further analysis shows that urine orotic acid levels are markedly elevated. The plasma citrulline level was normal. In this individual, where orotic acid levels are elevated, what urea enzyme is most likely deficient and what urea cycle enzyme is least likely to be impaired. So there are five enzymes that are associated with the urea cycle. And I think that arganosuccinate lyase is probably responsible for this breakdown in the urea cycle. So the elevated levels of orotic acid, so the citrulline levels, that's our big hint right here, is the citrulline levels are normal. So basically we wouldn't suspect any enzyme that comes before citrulline is produced in the cycle. So at least up to that point, it's normal. So the enzyme wouldn't be in that portion. And just looking at the cycle, so again, we have ornithine, and which is produced with ornithine aminotransferase, and then ornithine carbamyltransferase is what makes our citrulline. And since we have normal citrulline levels in this patient, we wouldn't suspect something that produces citrulline. But the next step in the cycle, arganosuccinate, that that is further along in the cycle and could have a deficiency there. And then also leading into our next question, arginine would then be our amino acid that becomes essential as a result of this deficiency. So our next question, a 47-year-old man presents with severe pain in his right great toe. He's unable to bear weight on his right foot and there is no apparent trauma. He reports the pain started last night and continued to increase in severity. Based on his physical exam and lab findings, he is diagnosed with gout. So gout can be caused by several different mechanisms. Blood samples are drawn. The results for the individual are listed below. So what is the best explanation for the presentation of gout in this individual? So just going down our choices here, is it the overproduction of purines? So there are genetic conditions that lead to an overproduction of purines, but they're extremely rare. And they're often associated with, and this is a long word and I'm probably going to mess it up, but phosphoribazole pyrophosphate synthetase. So you would basically need to test for a deficiency, some sort of defect with the gene that produces this enzyme in order to make this diagnosis. But again, it's fairly rare, so we wouldn't necessarily expect an overproduction of purines. Next, decreased salvage of purines. So there are a few diseases that we've gone over in this course that deal with decreased salvage of purines, such as Lesch-Nyhan syndrome. So this is due to a deficiency in that purine salvage pathway. So adenine phosphoribazole transferase is another enzyme whose deficiency can cause a decrease in salvage of purines, but you would need, again, more specific enzymatic tests to confirm this. Also, it's very unlikely, given the patient's age, that they're 47, that they would have decreased salvage of purines, as it's nearly always fatal in the first few decades of life for most individuals. So decreased urinary excretion of uric acid. So this is what the vast majority of gout cases involve. So we have a decreased excretion of uric acid and higher levels in the bloodstream. So our patient here has elevated levels of plasma in the uric acid, along with decreased uric acid concentration in the urine. So that means that somewhere along the way here, that uric acid is not able to clear itself out of the bloodstream through the urine as it normally should be able to. So this points to there being a decrease in urinary excretion, and that's likely our cause of this patient's gout. And then finally, after further discussion with the patient, he tells you that he recently switched from diet soda to regular soda because he read that fake sugar diet soda was bad for him, which is true. He says he drinks between 4 and 5 liters of soda a day. Yeah, so our hint here is that most sodas use fructose or high fructose corn syrup. And what is the potential impact of this dietary modification? So as we know from some of our earlier courses, basically fructose and high fructose corn syrup, fructose in particular, kind of skips a regulatory step in metabolism and everything. Is basically just stores fat much easier. So high fructose corn syrup is known to cause excess purine as well as skipping a step in metabolism, and thus, because it produces excess purine, it's definitely going to increase uric acid levels in the bloodstream, and as a result, this would exacerbate the patient's condition. They're already having trouble clearing uric acid from their bloodstream, as we saw by their laboratory test and our physical exam findings. Now we're basically just making that problem even worse by having them consume a food that is rich in purines.